speaker is Harshna Patel. She's going to talk about molecular simulation and virtual screening to investigate the structure and binding site of the influenza A virus nuclear export protein. So, Harshna, when you're ready, you can share your screen.
Good morning, everyone, or good afternoon, depending on where you are listening in from. I just want to say thank you to the panel and the organizers for giving me this opportunity. It's a pleasure to speak to you all today. In this talk, I'll give an overview of research that was carried out, which involved performing molecular dynamic simulations to model the influenza A nuclear export protein and to analyze its structure and potential binding sites. And I'll also share results of receptor-based virtual screening that was done to predict drug-like inhibitors that may bind to this protein. So I'm sure you are all familiar with this virus. It causes a contagious respiratory illness in humans and can easily transmit worldwide. But just to give you some background and put the work in context, I will give you some information to introduce the topic also. So there's 4 main types of influenza, A, B, C, and D. However, type A is the most common type. It's known to cause the worst disease in humans, and spread of type A has caused pandemics in the past, and annual epidemics continue to be reported. And this is partly due to the fact that this type A virus can display zoonosis, so it can cross-infect between different host species and form new subtypes, which transmit very quickly. The genome of influenza A viruses consists of 8 RNA segments, which encode at least 1 viral protein each. So around 17 proteins have been identified so far, most of which are shown in this figure here. Although only 10 are considered as the major flu proteins, which are found in all flu subtypes. The virus life cycle is quite complex, but can be summarized into 3 stages, starting with attachment, which involves the hemagglutinin surface protein, which attaches to sialic acid receptors on the host cell. This then allows the virus particle to internalize into the cell. It undergoes some structural changes. Once genome replication is complete and new proteins are synthesized, then finally the virus is released from the infected cell by neuraminidase cleavage. As you know, vaccines are available for some protection. However, this does not always offer long-term or extensive protection because the virus is able to mutate rapidly, and it does not protect against novel subtypes either. Antiviral treatments can be prescribed to reduce the severity of symptoms, and drugs that are currently licensed in the UK target the neuraminidase, the NA enzyme, and the endonuclease, which is part of the polymerase complex. However, the issue of antiviral resistance reoccurring or developing due to mutations in the binding sites of these antiviral inhibitors remains a concern. There is a lot of research and drug discovery efforts that are going on to target various other influenza proteins so that more treatment options can be available. But one which has not really been explored is the nuclear export protein, formerly known as the NS2 protein. So this protein is encoded by RNA segment 8. It's a small protein which exports viral RNA from the host cell nucleus to the cytoplasm after the genome is replicated. And this is an essential stage so that new proteins can be translated. And it does this by interacting with the viral M1 protein, which is shown in this diagram, and other host cell proteins, such as RAN-GTP and CRM1. There's currently no direct acting inhibitors identified against the nuclear export protein, and there is limited structural data available also. Only the C-terminal fragment, which is shown here as an experimentally resolved structure, is currently available in the protein data bank. So the aim of this work was to investigate the nuclear export protein structure through molecular modeling, as well as sequence conservation, and to predict and characterize potential binding sites. And then following this, we wanted to identify potential inhibitor molecules or drug-like compounds that may target this protein through virtual screening. So firstly, to account for the missing region that was not resolved experimentally, the ITASA protein structure prediction server was used to generate a complete full-length model using the experimental structure as a template. And then the region predicted ab initio by ITASA was then combined with the experimental structure to produce the final model. And this consisted of 2 pairs of parallel helices forming a 4-helix bundle connected by turns. Then to refine this model and improve the accuracy, a short energy minimization was performed, followed by equilibration, and then 3 replicate molecular dynamic simulations in solvent were run using GROMACS. The simulations were run for 100 nanoseconds at constant temperature and pressure. If you're unfamiliar with molecular dynamic simulations, it's an advanced technique that's used to simulate the behavior of molecules and can provide a dynamic view of the molecule over time. So with these simulations, position restraints were applied on the C-terminal. So the experimentally known part of the structure was kept restrained during the simulation. From the resulting simulation trajectories, root mean squared deviation analysis was done, and it was observed that the conformation of the N-terminal did not change significantly from the initial model, so it appeared to be rather stable. The backbone RMSD for the unrestrained N-terminal part relative to the starting structure is shown in this plot here, with each simulation in a different color. And then based on the RMSD plots, the trajectories were also analyzed to identify clusters of similar structures that were sampled during each simulation with an RMSD cutoff of 2 angstrom using the GROMOS method. The number of clusters produced during each simulation are listed here. And the central structure from the largest cluster, which is the most populated cluster, were all within 3 angstrom of each other. And these are shown in this figure here. So overall, this suggests that the conformations sampled during the simulations were very similar, but of course these results do depend on the cutoff distance that was used to determine cluster membership. And the root mean squared fluctuation of the N-terminal residues was also analyzed. And the results are shown in this figure here. The most flexible or fluctuating residues were found to be between positions 22 and 27 and 53 and 57, which correspond to the interhelical loop regions of the protein. So I mentioned earlier that emergence of antiviral drug resistance is a concern due to viral evolution. Therefore, the level of protein conservation was analyzed through multiple sequence alignment. So to do this, a large sequence data set was downloaded to get a good representation of sequence diversity. The multiple alignment was done and a scoring formula was applied to quantify conservation of each amino acid position. And then the conservation scores were manually mapped onto the protein structure by editing the structure file to illustrate the level of conservation. So here you can see the blue regions, which correspond to high conservation, and the red is the most variable regions. Overall, the results were in agreement with the notion that regions of functional importance remain highly conserved over time. Hence, the C-terminal part of the protein displayed high conservation so that binding interaction with M1 can occur. A few residues displayed 100% conservation, which indicate that they are the most, or probably the most resistant to change. And the least conserved residues were found in the interhelical loop regions, indicating that these areas should probably be avoided as drug targets. The 3 most representative structures from the clustering analysis were then extracted and used to predict the location of binding hotspots. So to do this, the FTMap algorithm was used, a web server, and this server applies different chemical probes to scan the surface of a protein and locate favorable binding regions. So the results showed that up to 10 binding hotspots were identified. These are shown in green on the structure. Common hotspots in the region between helix 2 and 4 were predicted for all 3 structures, but with different rankings assigned. Most of the hotspots were located in conserved areas of the protein, so within the blue regions, and several sites were also predicted fairly close together. One possibility for so many binding sites being predicted could be that other than the M1 interaction, several other NEP binding proteins have been discovered, although their binding site locations are unknown. So we think it's possible that their binding locations may coincide with predicted hotspots identified here. And because common hotspots in the hydrophobic region between helix 2 and 4 were predicted on all 3 structures, and they consisted of several highly conserved residues, this part of the protein was then selected as the target site for receptor-based virtual screening. So virtual screening is a technique used to screen lots of compounds by molecular docking to identify candidate molecules that may bind to a protein based on binding affinity. Just to try and improve the reliability of the predictions, a consensus virtual screening approach was used here in this work. So AutoDoc4 and AutoDoc-Vina were both used together. The National Cancer Institute Plated 2007 library, NCI library, was downloaded from the Zinc database, and it was filtered to eliminate compounds that may not display ideal drug-like properties, and to remove compounds which may be frequent hitters in virtual screening experiments. Over 1,000 compounds from the drug bank approved library were also screened to see if there are any approved drugs that may bind with a high affinity to the protein and could possibly be repurposed to target the NEP. So to illustrate the screening results, I've summarized the results in this graph here. The binding affinity distribution for the molecules from the NCI library are shown. You can see that the majority of compounds were found to bind with an affinity between around -4 kilocalorie per mole and -7 kilocalories per mole using both AutoDoc4 and AutoDoc-Vina. We would assume that the bulk of compounds binding in this range could be represented by compounds which have a common scaffold that display the same molecular interactions and therefore bind with a similar affinity to the target site. So overall, the predicted binding affinities were up to around -9 kilocalories per mole with the NCI compounds, and up to -8.3 kilocalories per mole with the drug bank compounds. The results from the screening using both software were combined. As I said, a consensus approach was used, and the top 100 or so molecules were inspected. These 2 compounds from the NCI library were identified as top hit molecules with the most similar binding scores from both software, and they also displayed a similar mode of binding. So you can see the predicted orientation and position of one of these compounds is very similar in the binding site. Many of the top compounds were found to bend around a cleft on the surface of the protein, so this may be a favorable binding region. And from the drug bank screening, we found that the top compounds were nandrolone phenyl propionate and estropipate. So these are also molecules that could be explored further. Results also showed that all of the top 10 compounds from the combined screening rank lists contained aromatic rings in their structure, but overall the structures are still very diverse. The specific molecular interactions, some of them are shown in these figures. So this compound specifically interacts with arginine 42 and alanine 102, which were highly conserved via hydrogen bonding, as well as other residues lining the target site through hydrophobic interactions.
Sorry,
this compound from...
Yeah, you have 3 minutes left.
Okay, thank you. So the compound from the drug bank library is held in place through hydrophobic interactions only with residues that surround the target site. So these compounds may be some of the best leads for experimental testing because they could bind with high affinity to conserved regions of this protein. So finally, I want to move on to current work that's building on what has been done. I don't have much data, unfortunately, to share on this at the moment, but we know this NEP does not function in isolation, and binding site properties can change upon forming protein-protein interactions. So I've now modeled the NEP and M1 complex using knowledge of the M1 binding site residues as constraints for protein docking. And the resulting complex is shown here with M1 in green. And we found that the binding site, which was identified on the monomer structure, is still accessible in this conformation, which is good. However, because conventional or classical MD simulations may not provide complete conformational sampling, more advanced methods can be used, like accelerated molecular dynamics, where a bias potential is added to raise the energy surface to accelerate simulation time. So these simulations are also being performed using NAMD to investigate the structure and potentially reveal new binding sites. CMD simulations are also being run, so this presents an opportunity to compare the performance of both techniques. I've been looking into principal component analysis, which may be more suitable to identify representative structures. And we may also look at sequence coevolution between these proteins to see if there are residue pairs that co-evolve and if that's got any influence on binding sites. So just to finish, overall, regions of the NEP protein were revealed in this work, which displayed high levels of conservation and ligand binding potential. Structural insights were revealed for the NEP monomer and in complex with M1, and the screening results could provide starting points for in vitro experiments or development of antiviral drugs that target the virus and may be effective long term. Finally, I just want to acknowledge my colleagues at the University of Hertfordshire and the University of Westminster, as well as the UK HEC Biosim, which have provided resources to carry out these simulations. If you're interested in working in the area of biomolecular modeling and simulations, I'm very welcome to work with you all and collaborate. Otherwise, thank you very much for listening, and I'm happy to take any questions.
Thank you, Herna, for this nice presentation. We have the first question is, did you explore one conformation's binding site, or how did you select the conformation?
Yeah, so the conformations were selected from the clustering analysis. Mm-hmm. So after the molecular dynamic simulations were performed, clustering was done to try and extract representative conformations from the simulation. And then once the most representative conformation was selected, the binding sites were predicted on each of these structures. So that's how we selected the final conformation through clustering analysis, and that's what we used then to predict the binding sites.
Okay. I also had a question. Did you try to build the model with AlphaFold 2, or the model of the monomer, or maybe the complex?
No, so I don't think AlphaFold does complex modeling at the moment. I think it's just monomer structures.
Yes, and also. Yes, you can do complex.
Oh, now. You can do complex. Okay, yeah, no, I hadn't tried or used ITASA at the time for the modeling, but AlphaFold definitely may be something to look into. But for the complex modeling, I use ClusPro for protein-protein docking, because the M1 structure is actually available as an experimental structure. But yeah, it was ITASA that I used for modeling the NEP.
It would be interesting to try. Another very quick question. Which method did you use to compute coevolution? Because viral sequences are very high conserved, and the most popular method cannot be used in that kind of alignment.
Okay, so the co-evolution analysis hasn't been done yet, but it's something I'm looking into, possibly PSI BLAST or another server. But no, I haven't actually done the co-evolution analysis. It's a step that's well, the next step, really.
Okay, thank you very much. So
thank you.
I will share my screen again. So
no. There were also some other questions that maybe, yeah, maybe Hershna would like to answer them in the chat.
Yes, I
later.
can do that.
Yeah. Okay. So have a look at the chat just to, yeah.
Yeah. Thank you.
Sorry for interrupting.
Thanks. Our following speaker is Darinka Pamela Duran Gutierrez from the Instituto Politécnico Nacional from Mexico. She's going to talk about in silico analysis of the impact of pathogenic variants in the interaction of natural substrate and a synthetic substrate with the human alpha hyaluronidase. So Darinka, you can share your screen when you're ready.
Hello, thanks for the opportunity of presenting this work. My name is Darinka Duran. I'm studying a PhD in biotechnology at Instituto Politécnico Nacional in Mexico City. And I will present some of the results obtained for an in silico analysis of the impact of pathogenic variants in the interaction of a natural substrate and a synthetic substrate with the human alpha hyaluronidase. Let me start by talking a little bit about alpha hyaluronidase, also called IDUA. This enzyme is a glycoside hydrolase structured by a Chimberell type domain where the enzyme's active site is located, a beta-centric domain, and a immunoglobulin-like domain, also called type 3 fibronectin-like domain. IDUA also has 6 N-glycosylation sites, one of which has been reported to be used by the enzyme as a substrate binding and catalytic model. This enzyme hydrolyzes the glycosidic bond between the terminal L-hyaluronic acid and the N-acetyl D-galactosamine of dermatan sulfate, and between the terminal L-hyaluronic acid and the D-glucosamine of epran sulfate. Both are glycosamine glycans composed of 200 to 300 disaccharide repeats, and they can be found as constituents of different tissues. When IDUA is not adequately expressed or has poor activity, a lysosomal storage pathology known as type 1 mucopolysaccharidosis develops. This disease is autosomal recessive and occurs in 1 in every 100,000 live newborns. Children with MPS I often have no signs or symptoms of the condition at birth. The characteristic features of the pathology usually appear within the first year of life for the severe form, while those with the attenuated form have milder features that develop later in childhood. For the mild and intermediate phenotypes, patients have symptoms including growth delay, aortic valvular disease, skeletal dysplasias, corneal clouding, and joint stiffness. In addition to having the symptoms, but in a more pronounced way, patients with a severe phenotype also have hepatosplenomegaly, mental retardation, hydrocephalus, and neurodegeneration. The diagnosis of MPS I is established according to the clinical signs and symptoms, determination of glycosamine glycans in biological fluids, and the evaluation of the enzymatic activity using the synthetic substrate for MUI, while the assessment of the phenotype is generally based on clinical signs and symptoms as well as the values of the enzymatic activity. Even though the synthetic substrate for MUI has been used for diagnosis as well as for the prediction of the MPS I phenotype, there are a significant number of variants that show no correlation between enzymatic activity and the phenotype developed by patients. Several reports have shown similar values of enzyme activity for patients with a mild and a severe phenotype. And there have also been cases in which even the activity reported for a patient with a severe phenotype is close to the one expected for a healthy individual. This occurs mainly with variants that are outside the active site. And well, since early initiation of treatment is more likely to improve clinical outcomes, and early diagnosis and accurate phenotype prediction are essential. Therefore, the establishment of a reliable and easy-to-use phenotype prediction method would be of great benefit. However, to achieve this, it is first needed to understand the differences between the interaction of natural substrates and synthetic substrate with IDUA variants. So to assess the impact of different variants on the binding of a natural substrate and a synthetic substrate, molecular dockings were performed with representative structures of IDUA wild type and variants located in the beta-centric domain and the immunoglobulin-like domain. The evaluated variants are related to the mild, intermediate, and severe phenotype of MPS I. protein 533 arginine is a relatively frequent missense mutation in the ADUA gene, while mutations in the residue serine 633, despite not being frequent, are of high interest as they have the characteristic of giving different phenotype. The incorporation of a leucine results in a mild phenotype, whereas incorporation of a tryptophan results in a severe phenotype. For molecular dynamic simulations of the wild type and mutant structures, the upper structure of IDUA was retrieved from the protein databank. 15 nanoseconds molecular dynamic run was performed, extracting 5 representative structures of IDUA at different times to be used in docking analysis. In the superposition of the representative structures of molecular dynamics, displacements of some of the non-catalytic loops, as well as some of the alpha helices that are part of the timber type domain are highlighted. To analyze interaction of IDUA with different ligands, 3 structures were used. The reported coordinates of the structure of a dermatan sulfate disaccharide, which is the endogenous substrate of IDUA, were obtained from the PubChem database. And the same disaccharide structure was used to design a tetrasaccharide, conserving bone distances and angles. Finally, the coordinates of the synthetic substrate for MUI were generated using the Biomodel Molecular Editor platform. The 3-dimensional structures of IDUA variants were designed using the Turnwheel platform, loading the wild IDUA structure and selecting the residue change to be incorporated, maintaining all the enzyme glycosylations. In these cases, also 5 representative structures of molecular dynamics were selected. And since this point in the superposition of the images, changes in the dynamics of the proteins were already appreciable, suggesting that this would have an impact on the interaction of the different ligands evaluated. Molecular dockings were carried out using the Autodock Tools program, where a grid box was set to define the search of the search region of potential binding sites for the ligand with the IDUA. An adjusted box with an appropriate X, Y, and Z size was placed around of the active site of the enzyme, and a Lamarckian genetic algorithm was used to get the docking conformations. Docking simulations carried out with Autodock Tools for a dermatan sulfate-derived tetrasaccharide allowed us to suggest that glycosamine glycans could bind to IDUA through a group that forms on the timber type domain to the left of the catalytic pocket, between the glycosylation sites of asparagine 372 and asparagine 415, which are shown in pink sticks. In the close-up of this region, it can be seen the surface of IDUA in which the substrate could bind to the enzyme, as well as the positioning of the terminal hydronic acid of the tetrasaccharide in the active site. The coordinates of the crystallographic hydronic acid were used as reference for an adequate interaction of our ligands in the active site. Also in this image, the surface of the substrate binding residues and catalytic residues are shown in purple and blue respectively. Interestingly, for this ligand, energetically favorable complexes were obtained only for some of the molecular dynamics structures of IDUA wild type and the variant causing the mild disease phenotype. These results agree with what was expected for the intermediate and severe MPS I phenotypes, where the catalytic activity of IDUA should be significantly lower, which would correspond to the protein 533 arginine and serine 633 tryptophan variants. For the dockings carried out with the dermatan sulfate disaccharide, a similar positioning of the ligand was obtained with the terminal hydronic acid located in the catalytic pocket and the next sugar to the left. Previously, reports have also highlighted the possibility that the dermatan and heparan sulfate chain stretches out to the open side of the catalytic pocket, but as far as we know, this is the first time that this is proved through docking simulations. In a close-up of the active site of IDUA wild type and the variants, some of the representative structures of molecular dynamics and the displacements of the binding residues and catalytic residues are highlighted. The results of the docking simulations with the disaccharide for the protein 533 arginine and serine 633 leucine variants showed complexes with binding energies similar to those observed for the wild-type IDUA complexes, at least for some of the structures extracted from the molecular dynamics, suggesting that the adequate structure of the active site of the enzyme may not be present permanently, reinforcing the idea that the structural dynamics of IDUA is indispensable for its function. Even for some structures obtained from the wild-type IDUA molecular dynamics, they do not allow an adequate active site formation. In the case of the assays carried out with the serine 633 tryptophan variant, only 2 structures allow the binding of the disaccharide, but with a binding energy of only half compared to the other 2 variants. Remarkably, it seems that for the binding of the synthetic substrate for MUI, the effect of the variants has lesser impact for the binding of this ligand. In most of the structures extracted from molecular dynamics, complexes favored energetically were obtained, and even in some dockings, the binding energy values were similar to those observed for some of the wild-type representative structures. These results fully agree with the proposal that the synthetic substrate is not capable of sensing changes in the IDUA that are outside the catalytic site of the enzyme, which explains the lack of correlation between the results of the enzyme activity and the phenotype presented by the patients. In summary, the calculated binding energies with the natural substrate are related to the phenotype given by the variant, where higher binding energy implies a lower affinity of the complex, which correlates with the worst phenotype. However, for the synthetic substrate, this correlation is lost, and we found higher affinities in the complexes of the variants associated with an intermediate and a severe phenotype compared to the complex of the variant associated with a mild phenotype. The results shown here support the proposal that the synthetic substrate is not capable of adequately sensing the catalytic activity of the pathogenic variants so that they can be correlated with a specific phenotype as a part of an early diagnosis of MPS I, since it could bind to any of the variants that are outside the active site with energy similar to those observed for the wild-type enzyme. But the obtained binding energies do not correlate either with the activity reported for each variant. It could have been expected that if similar binding energies were obtained for the wild-type and the variant structures, then the reported enzyme activity with the synthetic substrate would be similar. But in reality, this does not happen. IDUA enzyme activity decreased with the presence of the variants, as can be seen when comparing the reported enzyme activity with the reported activity for healthy individuals, either obtaining leukocytes or in dry blood drops. So the answer to this lack of correlation between binding energies and enzyme activity resides in the molecular dynamics of the enzyme. Different displacements on the Timberley type domain can be seen in the superposition of the representative structures extracted from molecular dynamics. In close-up to the active site, it becomes more evident the freezing that suffered the residues that are part of the catalytic pocket, as well as one of the alpha helixes of the Timberley type domain in the variant structures when compared with the wild-type structures. The clearest example of this is observed in histidine 91, which presented a wide displacement in the wild-type enzyme, while in the different variants, its displacements are reduced until it is completely frozen in the variant associated to the severe phenotype. In the case of the alpha helix of the wild-type structure, it also showed a greater displacement compared to that observed with the variants. Although these displacements do not seem to be so relevant for small substrates, such as the 4-MUI, in the case of a tetrasaccharide, it becomes evident that this dynamic will have an impact on the interaction of the glycosamine glycan chain, possibly helping to release the chain during enzyme catalysis in order to allow the next glycosamine glycan with a terminal hyaluronic acid to bind to the enzyme. So even though the synthetic substrate can bind to any variant located outside the active site, and adequate activity of IDUA will not depend solely on the availability of the catalytic pocket, but on the structural changes across the surface of IDUA given by their natural dynamics. As an early diagnosis of MPS I and the phenotype it might develop is crucial for an efficient therapeutic intervention. A better understanding of the regions of the enzyme that are important for binding to the natural substrate would allow the design of substrates that improve performance in supporting the diagnosis of the MPS I phenotype that patients could develop. According to this analysis, it is possible that a larger synthetic substrate might sense changes in the region where the glycosamine glycan chain is binding, allowing to correlate enzymatic activity with an MPS I phenotype. That's why actually we are working precisely on the design of a substrate that allowed us to correlate enzymatic activity with an accurate phenotype. And well, finally, I would like to thank to my team lab, as they have always been really supportive and hardworking. and we are at the Structural Biochemistry Laboratory in the Escuela Nacional de Medicina y Homeopatía. And well, if you have any questions, I'll be glad to answer to them.
Thanks so much, Darinka, for this nice presentation. So if you have any questions, you can write them down in the Q&A chat. So I will start with one maybe naive question. Did you try to quantify these differences in the movement of the lateral chain, as you show for the in your picture, that some mutant or some variants seems to freeze or to not allow the movement of lateral chains, and you showed the difference in different structural conformations. Did you try to quantify this movement?
Well, we obtained the RMSD and the RMSF for the, especially for non-catalytic loops, which are the ones that show differences in the displacements, as can be seen here. And it wasn't made for all the structures of the enzyme, only for some representative structures, especially the ones that was showing greater displacements in the wild-type enzyme and this freezing in the variants. I do not present those results, but we are expecting to publish them.
Okay, right. I think we don't have more questions. Thank you again, Darinka. I will move to our next speaker. Thanks. Our next speaker is Javiera Baez-Shara from Universidad de Telca. She's going to talk about the design of inhibitory peptides based on molecular modeling of the TRPM4 and KCTD5 interaction. So Javiera, when you're ready, you can share your screen.
Thank you very much to the organization. I'm grateful to be at this conference. Okay, today I'm going to tell you about our research, which is entitled Design of Inhibitory Peptides Based on Molecular Modeling of the TRPM4 and KCTD5 Interaction. Well, I can change. I don't know what happened. So, transient TRPM4 is a non-selective cation channel that conducts monovalent ions and is impermeable to divalent cations. Activation of TRPM4 can depolarize the membrane and activate voltage-gated sodium and potassium channels. Therefore, its function has been implicated in several important physiological processes, such as the Baylis effect in several arteries in response to stretch, the insulin secretion in pancreatic B cells, and the calcium-dependent immune response in T lymphocytes and dendritic cells. High expression of TRPM4 has been reported to be associated with pathologies that alter cytoskeletal rearrangement and cell migration, such as fibrosis and metastatic cancer in different cell lines. Okay. Each monomer has a three-layer architecture. The bottom layer consists on the nucleotide binding domain, the ankyrin repeat domain, and the C-terminal alpha helices, which is involved in channel assembly. The middle level is conceived of a helical domain, LHD, that contains 12 helices and forms the scaffold for multiple subunit interdomain interactions. The upper layer is formed for the transmembrane segments C0 to C6 and the TRP domain. So in 2020, KCTD5 was identified as a positive regulator that causes increased calcium sensitivity in TRPM4 and is involved in cell migration through channel regulation. TRPM4 KCTD5 expression was found to be elevated in malignant breast cancers with a poor prognosis, raising the possibility that coincident overexpression of KCTD5 may act synergistically to potentiate TRPM4 activity. Breast cancer is one of the main public health problems in the world. According to the official figures from the World Health Organization, it is estimated that 1 of every 12 women will develop breast cancer in her lifetime. Okay. Well, the KCTD family of protein is an emerging class of multi-domain and modular proteins. Its naming is due to the fact that the N-terminal of KCTD proteins and some voltage-gated potassium channels are homologous. A variety of functions has been identified for this domain, including transcriptional repression, cytoskeletal regulation, ion channel tetramerization, and closure, and proteobiquitination. Well, the structural characterization of this protein, the structural characterization of this protein, we can divide it in 3 parts, the hips, the torso, and the waist. And the structure in the N-terminal region has a relevant role in the homomeric assembly of the protein. Okay, well, TRPM4 and KCTD5 are mediated by protein-protein interactions. Protein-protein interactions, or PPIs, play an important role in the regulation of different signaling pathways and critical biological processes. However, they have considered an unviable class of drugs targets for small molecules whose science interface are often large and shallow, making them difficult to bind. Peptide-sized ligands have a great therapeutic potential as effective drugs, though their high specificity and activity, being larger in size, they are able to cover a large binding surface and are ideal candidates to these regions. These drugs derived from protein regions can be used as inhibitor or modulators and for mimic one of the participants in the interaction with the purpose of disrupting the interaction and investigating its biological consequences. The use of cyclic peptides has shown higher cell permeability and stability than linear peptides. Cyclization facilitates intramolecular hydrogen bonding within the ring structure, reducing the ability to form hydrogen bonds external to the molecule, and this decreases the polarity and increases the membrane permeability of the peptide compared as a acyclic cursor. I don't know if change. Okay. So, therefore, we propose to design peptides based on the protein-protein interaction regions of the TRPM KCTD5 complex. We used the sequence of human TRPM4 and KCTD5 to use sequence-based protein-protein interaction prediction servers. We use filters such as R residue pairs being in 3 prediction servers we use, not being in the transmembrane region, and having a SASTA score greater than 50%. In the image on the right, we can see the graph the results of the prediction servers. We assume that 100% is reached when the pair of residues is found in the 3 analyzed servers. In the figure below, we can see the residues that were finally chosen. Okay, for these residues, we use it as a reference for the grid in the docking processes. From the molecular docking prediction and the refinement, we obtain 2 models of interaction, R1 and R2 poses. With these models, we perform molecular dynamic simulations. The output was used to construct contact maps to see the regions of great interest. We measure the distance between the alpha carbons of the TRPM4 and KCTD5 residues across the molecular trajectory. Upon analysis of the interactions, it was found that the interaction regions in KCTD5 were similar in both systems. However, in TRPM4 interactions, we maintained in different segments of the nucleotide binding domain. In R1, the D chain of TRPM4 interacts with the E and F chain of the KCTD5, while in R2, the D chain of TRPM4 interacts with the F and I chains. So the underlying regions were used as the basis for designing the cyclic peptides. For this purpose, we extracted the peptide sequence and cyclized the peptide with the help of peptide modeling tools. I can change it. Okay. Well, we obtained 3 peptides, 2 based on the TRPM4 regions or R1 and R2 respectively, and 1 peptide based on KCTD5 on the interaction region. We use different cyclium techniques to achieve the final structure. Side to change to side chain, side tail to tail, head to tail for the different peptides. Well, this work is under development. Now that we have peptides, we will perform docking molecular dynamic simulations and free energy calculations to analyze if the peptide protein or protein-protein interaction is more stable. And we wanna use in vitro assays will be performed to see the uncoupling of the TRPM4 and KCTD5 complex upon interaction with the chosen peptides. Well, that was my presentation. I'm Javiera Baeza. Thank you so much for your attention, and thank you for my lab group, and Minicat, for this opportunity.
Thanks, Javiera, for this kind presentation. So, if you have a question, you can write it down in the chat. We're waiting for some questions about this project. So we are going to go far and test it in vitro. That's it.
Yes, I want to test that in vitro when we finish the peptides. When the energy.
Yeah, that's great. So I think we don't have questions.
Okay.
So thank you again.
Thank you.